Hello! Welcome to the
Hi.
Microbiology Journal Club. I'm Faz Alam. I'm a microbiologist. Here's my colleague Danny, who's also got a PhD in microbiology, and we're here to talk about microbiology papers.
Yeah, this is our on week where we dive into the figures. I think one of the things we connected about like doing this podcast, no, it's not a podcast, a YouTube channel, is that interest in like looking in the figures in detail,
Yeah,
right? Because you can hear the headlines and see the news things, and it tells you, oh, this great thing's happening, but until you dive into the figures, you don't really know like that nuanced uncertainty that comes with doing real
yeah.
science.
We're all about the data here. We're all about like understanding how things work and trying to have fun while we do it. Because I think even reading a paper and trying to understand how it was done can be an interesting activity. And that's well, part of the goal is trying to bring that activity to you, the audience.
Yeah. Yeah, so every other week we do like an in-depth discussion, and then on the alternate weeks, we look at the news headlines and look at the papers and try to choose something to discuss. And so this week we're talking about a paper called,
Yeah,
where
called
we got this?
SARS-CoV-2 cell entry depends on ACE2 and TMPRSS2 and is blocked by a clinically proven protease inhibitor.
Yeah, and this paper came out in April, so it's not a new paper by any means. Um, but it's one of those important papers that supports. I mean, by now probably many people have heard it in the news that like ACE2 is the receptor of
Mm,
SARS-CoV-2, right, of our novel coronavirus, and this would be the paper that came out to help support that.
yeah, and it's interesting because this paper has quite a lot of thing that's based on like basic science, what we understand about coronaviruses and the original SARS.
Yeah, I mean, it was funny. Like when we chose it, I remember saying like we said last week, oh, this is going to be like more of a biochemistry
Mm,
thing, right? Like we're not going to see anything clinical, but or like therapeutic wise. And I mean, that's not because they didn't try though,
mhm.
because I think that with the tools they had, they tried to make
Yeah,
claims, right, about potential clinical routes. But as we always say, like you're only as good as the models that you use, and in the models that they're using here, they're not using the virus itself,
mm,
right? They're not using, or I mean, I think maybe once they use actual SARS-CoV-2, but most of the time they're using something called a pseudotyped
mm.
virus, and and they're using cell cultures and stuff like that. That's not
Yeah,
those aren't models meant to show us things about disease, but they are models meant to show us interesting things about biochemistry and maybe set up for asking questions later about like how you could create an intervention around what you find.
yeah. I mean, I tend to be fairly biased against cell culture papers looking at treatments. I mean, I think we've only covered like one other one, which was looking at bat cells. Um,
Hmm.
but I think they're
But they
very
weren't looking at treatments there, right? They were looking just at like some
yeah.
evolutionary question.
When
Yeah.
they're good for actually looking at like kind of molecular interactions and understanding how viruses work. But when you
Hmm.
but like if you look at a treatment in a cell, there are so many other things, so many other steps along the way because you're not looking at a natural environment, but you are looking at an environment that can tell you a lot about how viruses work. And I think they do
Right.
delve into that. And there are areas where they try to make claims about treatments, but it's all very like um, mostly looking at the mechanisms and so this was like very early on where there was still and we still don't know very much about SARS-CoV-2. A lot of stuff that we know is based on what we know about the original SARS, and so this is one of the first papers to come out to basically try and confirm what we already know about SARS and try to figure out uh, whether that applies to SARS-CoV-2.
Totally. Yeah, throw up the graphical abstract. I think that's a good.
Yeah,
It has some info
yeah.
there.
So
Yeah, kind of like what did they know before going in this? They know there was this outbreak of classic SARS
yeah,
back in 2002. And from that, there's tons of research from that. And we've touched on that
mm,
before. We've seen a lot of it. And specifically like they have papers where they look at the crystal structure of of the
yeah,
receptor protein, the spike, right? This is what we've been talking about with all these vaccine candidates are for the spike protein. That's the protein that's gonna like bind to a receptor and like let the entry of the SARS go in. So they had crystal structures. The papers I picked out three and I put them in the Zotero if people want to follow along at some point.
yep.
But yeah, there's like Lee 2005. There's like a series of papers, some that are structural biology, where they look at a co-crystal structure, seeing this receptor against the spike protein. They do biochemistry, some of it, which is like what we're seeing here, but they also do some other ones. And I guess we can talk about that as we get into them, like the other ways of showing, right, whether something is a receptor or not. Because I think one of the cool things about, or one of the cool things about science, the great thing about science, right, and the important thing about science is you can show things one way, right, and you'll know it only through that one way that you showed the information. And so before we come to like a definitive statement about how something works, you have to see things through multiple ways. And so this paper presents like a line of evidence that tells us that this is the receptor, but there could be many ways to get that another line of evidence, right? There could be other experiments that could be designed to say like this is the receptor again, and we're only seeing this one snapshot right now.
And one of the things I quite liked about this paper is how it like clarified that receptor interaction and the fusion, because there are two distinct processes. Because we tend to think of the receptor binding as like kind of the lock and key hits, and then it's like, well, now what? And apparently, what the now what part is, so for most virus receptors, they need to bring that into fusion, so they need to like bring the cell membranes together so close that they kind of merge together. And this paper really delves into that process, because for coronaviruses, it's actually quite different from what you'd expect for normal viruses, because coronaviruses actually, in this case, are dependent on the host cell proteases to actually,
or some sort of protease processing, right? I mean, I think here they're definitely showing host cells, but again, they do things in cell culture here. So things are like, we know that these proteases exist in the body, and so inside our cell dish they also exist, and if we block them or whatever, we can see the effect. But there's always in the back of your head, I think we have to remember as scientists reading papers, right, that like that is in this system, right? And how does that actually relate to the actual animals or like actual humans? But yeah, like I don't think it's actually that uncommon to have protease processing, right? Even I'm pretty sure in influenza there's also pre-processing that happens to those fusion proteins. That's why they infect the club cells. I thought it was something because of the protease that's made by them,
right?
Some ability. Anyways, I feel like there was a reason for sometimes this is a reason for tissue tropism, right? Viruses, they have sometimes very specific places they infect. I mean, we saw maybe 2 weeks ago now, right, like a paper that did staining, and you could see like it's not the whole surface of the epithelium that gets infected, it's only specific cells, and that may be because different cells make different proteins, and viruses need things. They're so minimal, they need other things to support them.
Viewers, you want to refer to our long talks on the mouse models and hamster models, where they did really delve into like kind of those tissue tropisms in animals, which is and here we don't really get that sort of detail because they are almost monocultures of the same exact cell. And but they do a lot of work in multiple different cells, and
different cells might have differences between them, and that you can learn stuff from, right, the differences that you see. So I guess all that to say is before this paper came, right, there's this idea that classic SARS has a spike protein, right, that binds to ACE2, right, which is the receptor, and it requires cellular proteases to function, right? So all of that has been done, right? So, when we get to figures where they're like, oh, then they decide to investigate this, it's not out of the blue that they went down this discussion. They're basically saying, because it's so similar to SARS, right, can we confirm the things that we saw back then now?
Exactly, and that's what a lot of this paper does, and so I think we can bring it up to the first figure, which shows us the figure 1A, where they show the basic structure of the spike protein and divide it into two sections: S1, which is the attachment section, which is basically the focus of most of the vaccines, and then the S2, which is the fusion section, which is responsible for all the the interaction that brings the cell membranes together to cause them to fuse.
It's interesting. I feel like we've talked about S1 and S2 before, specifically in that vaccine sort of world, right? And when they're looking at convalescent serum, they often when they look at the neutralizing antibody panels, they say, is it S1 or S2 that they bind
Yeah.
to? With the idea that S1, and we've seen that with the epitope mapping. In S1, you have that receptor binding domain. Antibodies attached to there are neutralizing, or can be
Yeah.
neutralizing, often thought of as neutralizing as well. Yeah. And they do the alignment here in this A as well, so we can see the similarity between classic SARS and our novel coronavirus.
Yeah. Uh,
And there's some differences.
yeah.
There are some differences.
There are some definite differences there, but
Yeah, which is why study, this is a good argument for why the study is
hmm,
important, because, these differences, okay, I mean, just because like they're the same virus and they're related, there are still these differences, and we don't know if that's going to affect the way that we think
yeah.
about
I
them.
mean, we're like 60% related to bananas, but the differences are in really important locations. So like most of like the similarities are like basic metabolism, they're fine, but then other small subtle things can make really
Yeah.
massive differences. So we need to be kind of aware of that. That's why these sorts of things are necessary. There's this, there
Right.
are.
I like how you went with 60% bananas there, opposed to 98 chimpanzees.
I,
Like it's like even more
I
alien.
mean, I was going to go with chimpanzees, but I just realized looking outside, I've just been noticing the similarities. I think I'm very uncomfortable with how similar we are. I mean, I don't like.
Yeah, let's not emphasize that
Yeah, I
right
just
now.
yeah. So yeah, that brings us to the next figure, which is figure 1b, where this is where they start to establish the models that they'll be using for most of this paper. So in the top section, they've got this Western blot of the expression of these.
I don't think we've seen a Western blot before. This may be the first Western blot that we've looked
Yeah, I
at.
mean, I think the last one we saw was in one of our lost episodes where there was something really wrong with the Western blots, or yeah,
Oh yeah, I do remember that. Yeah, yeah, yeah. It was a supplementary Western
yeah.
blot. Yeah.
But yeah, so yeah, we don't. I'm quite glad because Western blots always they never look clean. Even the best Western blots can look a bit messy. So and to the point where I don't
I mean,
trust clean Western blots. I always assume because
sure.
my background is in research integrity as well. So I've just am conditioned to be suspicious of all Western blots I see. Um, so
Yeah, I mean, these are, I think, yeah, for people who are not familiar, it's a way of separating out proteins, right? So the proteins become these bands that we see, the dark bands. Those are pieces of
mm,
protein. And the reason why we can see them, why they darken, is there's usually some sort of antibody, something that recognizes that protein and then develops the color for
yeah,
us. So you use electricity to take your protein sample and stretch it out across a jelly, and then you use these antibodies. Well, you blot it, whatever.
yeah,
It's not super important, but you then use some antibodies to visualize those. And in these, so they're looking at the spike protein here because, again, previous knowledge knows that it gets
yeah.
cleaved from classic SARS. We know that there's a cleavage event that happens. Um, and are they using? Where are they getting this protein? Is this from already the pseudotype? They're
Oh,
expressing it
yeah,
on cells, I
so the
think.
top one they're expressing on H293 T cells. So these are like derivations of human kidney cells are designed to express lots of protein. So the top one is where they put
Right.
that kind of DNA into 293 cells and then got that to see whether it's expressed in them. So is it get created and how much of it is
Hmm.
created in relation to a protein that's already there. So ACTB, which is beta-actin, is a protein
Hmm.
that's everywhere in most cells. So mostly,
Yeah, the site is a cytoskeletal element,
yeah,
right? Cytoskeleton.
it is essential because without that we'd flop around. We wouldn't our cells wouldn't more so than usual.
More so than we
Yeah,
already
more
do.
so than we already do. But yeah, without that kind of cytoskeletal structure, a lot of internal cell organization wouldn't work. So it is essential to all cells, which is why it's used as a control because it's usually expressed at kind of the same level. So you can, so
Right. Yeah. So we want to see the band and the controls. When we're looking at Westerns, we want to see
yeah,
like there should be a band in those lanes that is the same across all of them to know that every lane like has the right,
yeah.
amounts of things that people put in and stuff like
Because
that.
otherwise you'll put in too few cells and think that your protein isn't being expressed, and then when you see the beta
Hmm.
actin, you see that's very faint as well. And they go, oh okay, and then you can actually use these days you can use technology to actually like quantify it now. Uh, but everyone wants to see the blots just to make sure.
Technology. Oh yeah, robot
Robot
eyes.
eyes, yeah. Image J is one.
Ah, yeah, that's a great
Yeah,
tool. Free too, free and open.
free open Fiji as well, great tool. Yeah, and the second part is pseudotype viruses. So, do you want to talk about how pseudotype work, or should I?
Yeah, yeah. So, I mean, they're going to use pseudotypes a lot. And, it could also be said as a criticism of the paper. And I think we may have touched on this before. You need BSL-3, I think, to work, or
Yeah,
BSL, you need a high biosafety level to work with actual infective
yeah,
SARS-CoV-2. So not everyone's going to have those facilities. And even if you do have those facilities, you don't always want to work in them all day.
yeah. You only have a few people trained to
um,
work in them, and they're very expensive because everyone wants to use BSL-3 facilities because that's where all the fun, amazing research happens. So there's a lot of
Right.
competition.
Yeah, I mean, we'll say like the more closely relevant research
Yeah,
as well, right? Like that's where they're infecting the macaques with
yeah,
the actual virus. That's where they're infecting mice with the actual virus. And so and so for this, it's a very common thing to use the pseudotyping system, which is, I guess, just shows you how much mastery we have over molecular biology. But
yeah,
the idea is that viruses, they package up. And actually, this is sort of a good primer as well when we're thinking about the adenovirus
yeah,
vaccine, right? How does it even get made,
yeah,
right?
exactly.
How do you make something like that? Well, viruses, they package up genomes inside of their little coats, right? The coats could be made of lots of different things. Usually there's signals in those genomes that help them get picked
Yeah,
up. So you can just put those signals in front of your favorite piece of DNA. And then and usually it's not done exactly like that. Like they actually take the genome of the virus and they replace things in it with the
yeah,
pieces. But anyways, you essentially have like a construct, a piece of DNA that is the virus for all intents and purposes, but has all the genes that you
yeah,
want. And so in this case, we put things in that we want, like reporters, things that make
yeah,
colors, maybe. We put in different receptors,
yeah,
right? And so BSV is not, has its own receptors, but through the process of pseudotyping, you can coat VSV particles with the receptor of your choosing.
yeah. And that's what they did here.
Yeah.
They added the spike protein in there, and they showed that these particles are expressing spike protein. In fact, it seems what they seem to have found is that they express the cleaved segment a lot more in the SARS-CoV-2 pseudotype virus compared to the
Mm-hmm.
original recipe SARS. Um,
Mm-hmm. Yeah, that's right. Like it seems like it's processed for whatever reason. It's probably cell type,
yeah,
right? I think it might speak to something like maybe the cell type is different, or the sensitivity to the protease is different that does the
yeah. I
cleavage.
think that might be it.
It's Oh yeah, I was just gonna say, but like, because it's different in comparison to original SARS. Like in the top panel of
Yeah,
B, where they're doing it in the cell lysate, there's the two
yeah.
bands, but in in the SARS-CoV-2, there's three bands.
Yeah, and well, it's weird because it seems like the SARS-CoV-2, like it's, well, I guess it might be because they might be slightly different heights or like, sorry, not sorry heights, but like,
Yeah, I think size-wise, they're definitely
and
different.
also there is this thing that happens with some proteins. So when you're running them through a gel, part of it is like dependent on like how heavy they are, but also like how they're conformed. So if your protein's really scrunched up, it'll travel through a gel a bit faster, whereas if it's like all nice and linearized, it'll travel a bit slower. So sometimes when you'll get like a protein that's the same size, but like could be slightly different. I'm not sure whether that's what's happened here or whether it could also be that there might be some other cleavage going on that means that it's appears in different sizes. So, but
For sure. Yeah, yeah, to explain the third band,
yeah,
right? It could either be like another cleavage event that we're just not focusing on right now. It could be some clumping sort
yeah,
of like action, right? A way of the protein like being stretched out. Like there's a version that's closed and there's a version that's open and they're just going
yeah.
through. Um,
I mean, my gut says that since it's in the cell, it's already being created and not fully conformed, whereas in the pseudotype virus, it'd be in its actual conformation because it's been already packaged.
for sure. Yeah, yeah. I do think that probably in the upper figure, it's some sort of artifact of it not being like made in viral
Yeah,
particles, right? Like Yeah, it's like something, all the pieces that are clumped in the membrane become another.
Yeah.
There's another formation. Yeah, it might not even be physiologically relevant, right? I think that's an important thing to remember, right? Is like as we move things out of the context of right actual virus
Mm.
and actual infection of host, we could be seeing things that are just
Yeah.
because.
Yeah, I think I
Yeah.
missed like what the author's explanation for this because I went because I couldn't find it in the paper. But again, that could be because at some point when you read a paper, you go blind a little blind. So if a viewer knows
Yeah.
what was going on here, then feel free to tweet me and tell me what the real answer is. Um,
Yeah, but the main thrust of the figure being that, you know, they can create these pseudotype viruses. So that opens up this whole research tool now for
yeah,
them, right? They have the ability to put the spike protein on their virus, the VSV, and inside of VSV, you can have a variety of different outputs, right? It doesn't have to be make more. It doesn't have to be like viruses make more of me, right? It can be show me a
yeah,
color, right? Tell me that you got in,
yeah,
and that's going to be one of their main assays to show us right that fusion
yeah.
happens or fusion
They
doesn't
also
happen.
they put in the green fluorescent protein in their VSV virus, so they can actually kind of get check by looking at the color, see whether the virus is
Mm-hmm.
infected or not.
Yeah, yeah, and I guess maybe it's important to remember too for people who aren't so familiar with molecular biology, they're putting that sequence of that GFP in it. So, right, that doesn't glow by itself. It has to get into a cell, and the cell has to be, you know, co-opted to make it in order to see the green. And so, the output of looking at these assays would be green
Yes,
cells.
exactly.
Yeah.
So the next step of the is where they actually infect an entire panel of different cells, which I really like. I think this is
Yeah.
absolutely essential for any cell line study, uh,
Hmm.
because
I mean, that's also the strength of cell line studies. Like, there's all these cells that we have, right? You don't have to boot up the whole animal. We have them in little vials sitting in the freezer, right? And you can capture all this biochemical complexity, right, between the different species, in your incubator,
yeah.
in your lab.
So I mean, they got like a bunch of so
Yeah,
I'll just go through some of the lists of the human cell lines because they got
yeah.
like eight.
So this is the control
Yeah,
figure
this is
first,
control
right?
figure. So this is with the VSV expressing its actual native VSV kind of receptor to fusion cells.
Which fuses to
Yeah,
everything.
it's actually a really good one. I don't know why we're not frightened of VSV now, but um um but yeah. So again, this so we've got A549s, which is alveoli cells that have been taken from an adenocarcinoma alveoli, so lung cells. We've got BEAS2V, which is non-tumorigenic lung epithelial cell line. So they're a lung epithelial cell line that tends not to create tumors. See
Hmm.
CALU3, which is a lung cancer cell line. We've got H1299, which is another human lung non-small cell carcinoma cell line. We've got C293T, which is a human kidney embryo cell line, which was transfected to make it express certain things that make it express more protein. Got
Hmm.
HER7, which is a hepatocellular carcinoma cell line, so that's basically like a liver cell line. We've got CACO2 cells, which is a colorectal adenocarcinoma virus. MRC5, which is from lung fibroblasts derived from an embryonic cell line. So
Hmm. So all human cell line. This first set is all
all
humans.
humans,
They have our cute little male female washroom
yep,
symbols at the top. And it seems like a lot of them are
yeah,
lung, right? Because there's a lot. I mean, we know that they infect the lung. This is like we hope to see right in our
yeah,
SARS-CoV-2 panel some of those things, but throw in some other ones in there too because you know we know some things about SARS-CoV. We might want to suspect some things about SARS-CoV-2 infecting other places as
exactly.
well. And I think this is here where this is where all the meaning is
Mm,
coming from, right? Is this list of cells
yeah,
because while they are just cell lines, right? They're the first step in like drawing us to one sort of conclusion or another,
yeah,
right? Oh, tropism for these cell types, or like works here but not here. And then of course these are well studied
yep,
lines, and so people know like the inventory of proteins that are associated with
yeah.
them. And so like that's also where the meaning is coming from.
And they do multiple ones because obviously, because the thing about cell lines is sometimes they can be slightly different from what you see the host, and but the fact that they all diverge in different ways. So if you look at lots of them, you can actually build up a picture about what happened, what might have happened, because they vary a lot. So having lots of them means you're almost like doing extra replicates and getting more variation, and seeing, and with that variation,
Yep.
if you can see something that kind of is maintained.
The same, yeah, is maintained, then it's more likely that it's something that could be physiologically real, or and at least you can narrow it down as
Yeah.
well, right?
But then they did a bunch of other animals. So they've got like Vero, which we've talked about before. It's from the African green monkey. They've got pig kidney cells. We've got bovine kidney cells and canine kidney cells. We've got two bat cell lines. One from the horseshoe bat and another one from the Dorbenton's bat. So horseshoe bat is associated with SARS-CoV, and Dorbenton's bat tends to be associated with Ebola. So I mean, in terms of cell lines, so this is based on brief like Google of where these cells are used. And then you've got BHK21, which are hamster kidney cells, and NAH73, which is mouse cells.
Yeah.
So
have you used a lot of these cell lines before?
I have not used any of these cell lines.
Oh, really?
Yeah, no.
I've used A549s. I've used 293Ts, 3T3.
Yeah.
I've seen MDCKs before.
Yeah.
Oh, and CACO2s. This is like, this is very much like somebody's fridge of things.
Oh, yeah. I mean, yeah. Again, I have no experience with cell lines, which is why I try to read up on them as much as possible, because I know that this is my weak point. Because again, I'm biased against cell lines. So we use primary cells in my previous studies. So again, like,
gotcha.
I was associated with the hospital, so we actually had access to patients who we could get cells from, whereas actual real like basic scientists don't necessarily have that kind of access to stuff. So
it's true.
Yeah.
And at the same time, it's like you use what you have around, right? Like these are well-established things. Research is known. Like it's not that the information that's going to come from them is like bad, right? It's just that it has a different set of limitations, right? Everything has a set of limitations.
And you can actually, I think you can actually learn some things that you might not be able to learn just because of this. Sometimes you can learn things when things are different. Anyway, so we've looked at this control.
Okay, so yeah, VSV works.
Yeah, but now we look at the actual original version SARS.
Yeah.
So
again, pseudotypes, right?
Yeah.
Because they figured it out. So.
Yeah. So this is the pseudotype, the amount of times they get entry. So this is looking at luminescence and based off of the background
over an uninfected control that has some background glowing.
Yep. And yeah, we can see.
And so in all those lung cells, we're seeing infection.
Yep, in all those lung cells.
Sort of entry.
Apart from MRC5, but again, that kind of speaks of, which is also a lung cell, but it's a fetal, like, basal stem cell line, so it's not necessarily going to produce the same thing. But there is that diversity there that I was talking about, which is actually a good thing to have.
Yeah.
And then we've got, like, Vero and canine kidney cells, which seem to both...
Yeah. I think the other thing to remember too about cell lines is even though they come from the spot in the body, it doesn't really mean that they resemble the cells in that spot in the body either, right? Like you mentioned, for some of these, they're cancer, right? They have carcinoma after them because they were isolated from cancers.
Yes.
And like since then they've been passaged many times. So there's another part there. It's like yes, they're related to the body part, but there's also abstraction from that. They're also just like their own thing.
Because when something has cancer, sometimes it can like spatter out lots of different proteins, and that's part of what makes cancers dangerous sometimes, because it can end up evolving a new strategy to avoid your immune system just through the way they mismanage your proteins.
Right. Right. All right, let's see SARS-CoV-2.
Yeah, the star of the... So again, actually a very similar pattern, except it's less infectious to dog cells, so that's good for those cells. I can't really say whether it's good for dogs, but...
We've saved dog kidney cell line.
Yeah, I saved just about that last minute where I went, oh yeah, it's just so good for those cells, but no.
Yeah, but the pattern is the same with that MRC5. Like, there's something about that cell line that isn't jiving with the known receptor. Yeah. But because the profile looks the same, that's already along the line of the hypothesis that it could be the same
Yeah,
receptor, right? Like we're moving closer in that
exactly.
direction.
And then that brings us to we've got like well that shows all the whole figure. Sorry. So
Yeah.
the next one,
I mean, it's good. Now we can really see the comparison, right? How the profiles look the
yeah, we
same.
can really see that profile. And then there's a couple of like figures in the supplementary section which also kind of support these sorts of things. So again, you can see that the MRC5 is not caring about anything, but you can also see like kind of various
Mm.
correlations between kind of side by side, whether between the original recipe SARS and Zinger SARS, which is the recent one. Uh,
The spicy one.
Spicy, yeah.
Yeah.
Uh,
Yeah, I think also what's nice to see here, what I enjoy is like seeing the baseline,
yeah,
seeing where the baseline actually is in relation. Because it's one thing to say fold increase over
yeah,
baseline, but like I like seeing baseline as well, and it just re puts the bars, same bars, right? They're the
yeah,
same data points. They're just shown in a different way for us.
yeah. And then the next one is 3A, which is where they have a relatedness map. So a f-
Hmm.
I want to say phylogeny, but when you're looking at just a single protein, isn't actually proper phylogenetics because with phylogenetics they do all sorts of other complicated like looking at various other controls. This is just looking basically a comparison of the similarity of the spike proteins and the specific
Hmm.
like
But similar to like a graph that we would see in a proper phylogenetic
yeah,
study, they are showing us like there's these numbers, right? They put these little numbers by each node,
yeah,
and that's because when they run these algorithms, they run them like many
yeah,
times, and sometimes because computers they're doing all these pairwise comparisons. Actually, not every computer is doing very well. Sometimes humans can do this better than
yeah,
computers can, right? There's a game. There's one of those like fold it style
yes,
games, crowdsourced games that people do them. But anyways, all that to say, the algorithm runs many different times. And then it gives us kind of like I think these are like called bootstrap percentages or something like
yeah,
that, right? It's the probability in all the trees that they ran that node is what it is.
yeah,
Right, that node exists, the difference between. So like 100 is meaning like, okay, right, based on what we know, the information we have in the computer algorithm, right, it's always giving us these two things are split here. The node exists here. But things like 77, like those could be compressed,
yeah,
right? Like, it's pretty good. It's like B plus, right? B plus node separation. But there's a chance that those are switched,
yeah,
right? There's a chance they're switched, or they're just a branch. It's not a node at all.
yeah.
And so that just gives you some uncertainty in here.
I mean, I guess the main point is try and show like how similar this family of coronaviruses are, and also point out like how common it is for ACE2 to like evolve in different strains. So
Yeah. Yeah. Yeah, so in some of in a bunch of these coronavirus proteins, there isn't ACE2
yeah,
usage, right? Which kind of makes sense because not all of these are human
yeah.
infective SARS. Yeah.
So lots of these shouldn't.
I actually maybe wait, are they human infective
So
SARS? Are they bat
most of them are
SARS?
seem to be the human infected SARS are in like orange, and I
Right. They'll say human if they're yeah, because they call them SARS-like
yeah,
in others. Also, naming conventions might not be the most informative for all of
yeah.
these.
And most of the time they're just based on where they're discovered and like
Right.
and they've added red
Yeah.
dots to like some various like bat coronaviruses that seem to have ACE2
Yeah.
binding properties. Um, but yeah, I mean these trees again. This part of it was with this early on. I mean it's based on like how good the data is you're putting into them, and how many other things you're comparing. So I don't want to like
Hmm.
we don't want to say that we now know the origin of SARS because this is just one small snippet, but it does tell us something about
Right.
how the spike protein relates to the other spike proteins.
Exactly. Yeah. Oh, the other thing I guess to remember about these is that there's always something, they always need something out group, right? Zaria, Bat, Kobe.
Yes.
They always try to put one thing that's like very unlike all the rest just to just to make sure um because this is just a comparison within the group that they are
Hmm.
testing, right? And so if you didn't have something that was wildly different, you could get like a lot more uncertainty in some of the nodes, right?
Right.
Because when you're looking at a small set of differences, it's hard to say like what is more the most different. And so they need to include something like
Yeah.
that. When you see these, you want to see the outgroup
Yeah.
in there.
You don't want to overfit and like end up finding like tiny,
Mm-hmm.
tiny differences that don't make any difference, but they become expanded. Whereas having something that's completely different
Mm-hmm.
helps to ground things slightly.
Yeah.
Because with these graphs, it's all about comparisons, and so having that control is
Right.
very important. Um,
And it's kind of nice. Already you can see some patterns overlaid on it, right?
hmm,
Like they do the geographical location of where they come from, and that's represented in the comparison,
yeah,
right? And you can like yeah, the nodes split out. And I would say like from this because you can see that they put it in gold for us, right?
yeah,
Our
yeah,
our Wuhan CoV2. Um, so it is actually there's a significant separation between classic SARS, right?
yeah,
And I guess what I presume what these the red dot ones are,
yeah,
like the strains, those are the thought to be progenitors of classic SARS.
yeah,
Yeah, so like you can also even from this you're sort of seeing like, oh those are also split from each other, right? By that 75
yeah,
node, right? Both have the signature of being from Asia. Yeah, but they might be different. So again, reasoning for why this study is important,
yeah,
but why it's likely that there's good reason to also believe that it's using the same
yeah.
receptor.
And in figure 3B, I think it is. Yeah, they. Oh
Mm-hmm.
no, not 3B. This is 2B. Sorry, where they look at the actual homology in the ACE2 binding kind of section between this SARS and the original recipe SARS, along with like a couple of versions of the virus that don't have that receptor, just to kind of isolate what are the important like kind of structures in that receptor binding, like kind of well, you call it
Mm-hmm.
cassette, but motif that actually, uh,
Yeah.
yeah, which I think,
Yeah, because they know the ones that don't bind ACE2 and the ones that do,
yeah,
right? So that would be a way to see the exact amino acids that could be important. And they're seeing similarity between our oh well they're seeing mixed they're seeing
yeah, the
mixed.
thing.
And actually the funny thing is that they could go deeper into
Mm,
this, right, a little bit. And I mean, how did they know that these things bind? And again, it's because they did the crystal
mm.
structure for the classic SARS, right? That's why they know these.
Yeah, I mean it's interesting because I mean if theoretically they could have like had a version of the protein that binds ACE2 in a completely different way, and that in itself would be quite interesting
Hmm.
as well. So confirming
Absolutely. Yeah.
that those similarities in binding are the same is actually can almost be a surprise. Um,
Sure.
yeah. So now moving to 3A, which is looking at the pseudotype virus expressing different spikes, infecting in different cell lines that are expressing
Hmm.
different.
Yeah, so along the bottom is the different pseudotype
Yep.
viruses. We have our classic, we have our current
Yep.
current contender.
And
Um,
we've got
what's
added in. We've got two other viruses. So you've got like two two
MERS?
nine.
MERS is a different
Yeah,
receptor.
MERS is
Yeah.
DPP4 and 229ES attaches to HAPN. So these are two separate viruses that are also coronaviruses in that family that attach
Coronaviruses.
via different receptors. And this is done in
Mhm.
hamster kidney cells, which don't necessarily have these receptors and aren't really very good at the whole infection thing. I think we saw that in a previous graph.
Right.
Um, so again, they see that the VSVG that's our positive control that infects everything.
Yeah.
Um,
There are
SARS,
cells in these dishes, and the cells respond to the reporter. They
yep,
become green. Yeah.
and the human ACE2 and bat ACE2 are both attached by both like original SARS and now SARS-CoV-2,
Hmm.
and the other two coronaviruses seem to be confined to their specific entry protein.
Hmm.
So
Hmm.
again, that's quite interesting.
This is the money making figure, right?
Yeah,
At least for part of the title, right?
yeah, exactly.
Mhm.
This is what we need to confirm that oh yeah, so SARS-2 and SARS-CoV both like enter cells via the same way they react to human ACE2 and bat ACE2.
Right,
So we kind of feel like we know what we're dealing with here with SARS-2.
right.
And
And this gives you the sufficient case,
yes,
right? Right? If
yeah,
these things are together, it will get you in, but not that it can't get in another way. We don't
yeah,
know about that part.
yeah, exactly.
And again,
Um,
because we've only reconstituted as well, like one protein on the pseudotype surface. So this is only talking about this interaction, not necessarily the interaction of the viral particle itself. There's some other
yeah,
proteins on the surface that could modulate
yeah,
this. Hmm.
yeah. So this very study is focused on spike, which is
Hmm.
not surprising because spike is a very important protein.
Yeah. Hmm.
And then with the on figure 3b, they look into anti-ACE2 antibodies and whether they disrupt that kind of interaction between SARS and its receptor.
Hmm.
Um, and they find I mean,
It
because you'd
does.
hope that it does if that's an actual genuine binding, and
Yes.
they find that it does in like this dose dependent manner. So they have like a low dose of 2 micrograms per mil, and then a high dose of 20 micrograms per mil, and that graph goes like right down. The more you of this antibody add, the more this reaction gets inhibited. So
Hmm. Yeah,
that's
which is very consistent with the idea that something real is right. It's not just some
yeah,
fluke of what's being added, right? It's the thing that I am adding is creating this effect.
yeah. You've got that extra line of evidence
Hmm. Hmm.
to back that up. And then finally, they use real version SARS-CoV-2 to test this interaction.
Ah, the only figure I think, right, in which they use real.
I think this is they use maybe one other figure at the
Yeah.
end, but this is like the first one where we've got the real. So because we're just playing around with this pseudotype virus, we don't have the real one, but this one uses like our this real version of the SARS-CoV-2 that was isolated from Frankfurt, I think,
Hmm.
and then they measure it using RNA. Sorry, so what is
RT-PCR,
RT-PCR?
yeah,
Yes,
genome equivalence.
yes, genome
Genome.
equivalence, and yeah, they kind of see that same sort of thing where they they have like cells that are expressing human ACE2 versus control,
Hmm.
and then measuring it at different times, and yeah, it infects the cells that have ACE2 compared
Yeah.
with the controls that don't have ACE2.
Yeah,
And what?
I mean, I like this. This is very much like this again. To me, this is the money-making figure for at least part of the title, right? The lines of evidence that show us that this is the receptor, sufficient to be the receptor.
Yeah, yeah, that's really important to establish,
Hmm.
and then that brings us to S3A. So this is where they start moving on to the second part, the membrane fusion part of the cell exercise. So um, there are two like cellular proteases that they talk about when in relation to SARS-CoV-2 being tr- the spike protein being cleaved and forming that fusion conformation.
Hmm.
So there's TMPRSS2 and this second one called CATBL.
Hmm.
So
And they're
CATBL,
both ones in the endosome, right? That's
yeah,
so the idea is that, you know, for if people have any cellular biology background, it's like the cells do endocytosis first to put in like some of the extracellular stuff with viruses coating the outside of that piece of membrane, right? Comes in, it becomes a endosome, a vesicle inside of the
yep,
cell, and then in that environment is when the virus then escapes out into the cell, into the cytoplasm.
yeah. So like when cells are like eating or drinking, they like take things in by like grabbing and making little bubbles and then
Hmm.
digesting it,
Hmm.
itty bitty wise. And
Hmm.
so when those cell endosomes get again, they acidify and they kind of release acid to like break down certain compounds.
Hmm.
And one of the things that is activated when that happens is CATBL. So what
Yes.
they did was they gave these cells ammonium hydroxide, which is kind of very alkaline product that prevents these endosomes being acidified. And if those endosomes stop being acidified, then that CATBL stops working. So if like SARS-CoV-2 relies solely on that, then just by adding that ammonium hydroxide would stop it from being absorbed.
Right.
And and from what we see, so they
But
compare
wait, this
against
is this where they added? This is not where they added ammonium hydroxide, is it? A?
it's S3A. So yeah,
Oh yeah,
they
yeah.
yeah. So it's complicated because the way they talk about these S3 and then they talk about 4A. Um, So yeah, they added like also Nipah virus as a control because that isn't affected by the
Hmm.
alkalinization of of the endosomes. So and they find that in 293,
Right.
which are expressing ACE2, that the SARS-CoV-2 don't infect. The pseudotypes don't. Sorry, the pseudotype
Right.
virus expressing spike does not get into
Yeah.
the cell, and they find something similar with CACO. Well, something similar to CACO2, but there's still some background there,
Hmm.
so indicating that there is some other protein that might be having an effect in those cells,
Right.
which we presume must be TMPRSS2. Which
Because that one's
bring,
not affected by acidity in the same way.
yeah, yeah, that's right. So I'm now on S3B, which is looking again at 293 T and ACE2, looking
Hmm.
at
But now they've added
um,
in a new inhibitor or two new inhibitors.
yeah, two new inhibitors. So they've got this one inhibitor called Camostat. Sorry, I'm
Yeah,
mispronouncing. Yeah,
Camostat.
Camostat and E64D. So Camostat inhibits TMPRSS2, and Camostat. Uh, no, Camostat. Yeah, sorry. Oh, I got confused there. Camostat inhibits TMPRSS2, and
Yeah.
CATBL is inhibited by E64D.
E,
Uh,
yeah, E64D is CATBL,
CATBL, yeah, I got
and Camostat is TMPRSS2.
PRSS2.
Yeah.
Right. Yeah.
Yeah. But anyways, they're basically they it's a drug that they already know, right? They have some big um like panel like previously known
Yeah.
inhibitors, right? Like, people have been trying to make drugs for a very long time. Every almost
Yeah.
so many things have been considered as targets that even in this case where we just have like a biology question, like we have inhibitors, pharmacologic inhibitors, to start asking like if we block it, will it work?
Yeah.
Will it still work?
And I think TMPRSS2 is associated with some fair and some number of cancers, and I think that's why we have some
That's why we
inhibitors
have
for it.
drugs here. Yep. Hmm.
Yeah. I mean, the great thing about cancer like research is you're not just funding like a cure for cancer, you're funding a great understanding of how cells work,
Yeah, a
which
lot
is
of cellular
why
biology research.
yeah, which is why this cancer drug suddenly can be used for looking at investigating infection.
Hmm.
So
Hmm.
everything is connected. In my conspiracy whiteboard, and so they look at like so the thing about the 292. So they got one which has ACE2, but I think that one doesn't have the TMPRSS2, if I recall correctly. Um,
Yeah,
so
the CACO2 cells, right?
yeah,
Yeah.
so yeah, the CACO2 cells. So it kind of suggests that. So when you see that kind of difference in camostat with those cells compared to the other ones, it suggests that that's having an effect in those cells. So that's
Hmm.
explaining why that the previous figure there was something there in the CACO cells, that there is that TMPRSS is in there, is in there. So therefore, it's having an
Yeah.
effect. So these two
Camo
like
stat is blocking. Yeah, camostat is blocking TMPRSS2.
TMPRSS2.
And in the 239 T cells, it's not in that cell line, it's not responsible
Yeah. So
for the entry.
that's why I have these
Something
big,
else.
beautiful blue bars in the top figure B because the camostat should be having an effect, but it doesn't because TMPR SS2 is
Yeah.
blocked by camostat. Um, we're going to be saying that a lot. We're going to have to remind ourselves about that because again, these
Yeah.
sorts of things
No,
can
it's
get.
it's not blocked by camostat, right?
Oh, wait, it is a
No,
TMPR
no, no.
SS2 is
The
blocked.
reason, like in figure B, the top one, 293 T cells,
Hmm.
the camostat is blocking the activity of TMPRSS2, and we see that it has 100% of the control um,
Oh
entry.
no, I think that's so. I think
Or is
that
that the opposite?
it's the opposite because TMPRSS2 helps the spike cleave to allow fusion with the cells. So if you block TMPRSS2, that spike can't fuse.
Right.
So that's why it's in blue.
I
Uh,
see.
yeah, it's weird. And I think that brings us to figure
Figure
4A.
5. Oh, yeah, figure
For
4A.
yeah, I know it's complicated
It's the
to.
same. Yeah, I see.
So
Because in the Vero cells, it's also it's the same that we saw in the A549 cells.
yeah,
The
uh,
same pattern.
yeah, because they've got like two versions of the Vero cells. One that has no TMPRSS2 and one that's had TMPRSS2 added. And then we
Hmm.
can see like that same pattern that we see with the 293T and the CACO cells, where where there is no TMPRSS2, there that chemostat doesn't seem to work. But on the other conditions, actually E64 E64D does work. So actually, and using them both seems to work every time. So
Yeah. Oh, so maybe Camostat doesn't block TMPRSS2? What?
uh, it, it it does block TMPRSS2, um, but so
Sorry. Yeah, I feel like I got a bit mixed up
yeah,
there.
it's the logic of these can get because if it does block TMPRSS2, then the viruses don't fuse and you get a
Yeah.
much lower value on the bars.
Hmm. Well, that's why the double. I understand like the double inhibitor is blocking all the potential, right? Things
Yeah,
that could cleave, and
yeah,
that gives us down here. So then they add in more TMPRSS2 or into the Vera cells, and now right, and now they get a response even with this inhibitor.
yeah. And the interesting thing is when they block E64D in those cells, that doesn't seem to work as well as when TM.
Hmm.
So yeah, I mean, yeah, it's one of those things where I have to read through this several times so I can be clear in my head what I'm going to say. Um, and it's
Yeah,
difficult
sorry. I guess
because
obviously I didn't think about that they used cells that then added TMPR, so that's why I just
it's
was a bit.
yeah
Yeah.
these I mean that's part of the fun of like because these are molecular pathways and trying to pick out which is which because we're trying knocking down the function of each one and having to keep the negatives and the positives all in your head at once can actually be
Right.
very complicated.
And
So
the
like
but the way that they're knocking it down too, it's like it's through these pharmacologic interventions,
yes,
which may not be specific for one protein itself.
yeah,
So that's why they add some in, I think. That's to help that. Yeah. Mo, sorry, I missed that when I was reading this.
uh, yeah. I mean, if other people in the comments, if you can, I mean, I always find it helpful to draw a diagram on a whiteboard to try and figure out these sorts of things.
Yeah.
But uh, but yeah, again, it's one of those diagrams that makes sense to me as I'm drawing it. Then the moment I look back at it, I'm like, what did I do? Why did I do this?
So in
Uh,
B,
yep. In B, we had. Oh, this is an incredibly complicated one because this took me a while to figure out because they have cells that are expressing TMPRSS2 and they've added E64D. So let me
hmm
see what my notes say on that. So the idea is that the adding of TMPRSS2 can negate any effect of E64D. So it's essentially like you can knock down the effect of CATBL, but you can still get the SARS-CoV-2 viruses. The spike protein can still fuse if you add in TMPRSS2. So think
after the fact,
yeah. So
because
effectively
when they used it before, they were preloading it with they incubate the cells first with some of this.
yes.
Yeah.
So this is just to further like boost that kind of idea that TMPRSS2 is a really important protein in the fusion process. But I
Hmm.
do kind of feel like you could have reversed this and did it the same way, but with the CATBL, and you might be able to see a similar sort of thing happening.
Sure.
Uh, yeah, let's see what else. So I think that's got. I think we're going to figure S4C where they look at the dosage of camostat and
Hmm.
that effect on pseudotype entry. So again, we've got VSVG, which is just infecting everything, and we see like with MERS and SARS-2S, they all have like this dose effect. So I think this kind of speaks to the fact like how similar coronaviruses are. Whereas
Yeah.
MERS, it has a
They need
different
some
like,
sort of processing. They have a similar pre-processing step from a host protein.
yeah. And firstly, it's quite interesting from an evolutionary perspective that they all have this need for that kind of processing. But also from a drug perspective, you're thinking, well, actually, that means that this kind of treatment could not just help SARS-CoV and SARS-CoV-2, but also MERS, other coronaviruses, and perhaps even whatever future coronavirus comes out that will surprise us in the future.
Hmm.
So again, this is the money maker as a
That's what you. This is what's money making for you. That
yeah, this is what's money making for me. I'm like, man, I'm gonna invest in this drug, and it'll protect against all coronaviruses.
Yeah, I guess this is the other part of the title, right? Like
Yeah,
this is the thing that does the inhibiting a potential therapeutic, although only
yeah,
showed in cell lines.
yeah. Only in cell lines.
Yeah.
So that's the bit I'd say under my breath to investors. It was great in cell lines. And that brings us
So
to
yeah, they do it again with the full virus then
yeah.
to
They
show that the
also
camista has an effect. Oh yeah,
yeah.
sorry.
Oh yeah, and in sorry, I want to jump to like because the S3C looks at the side effects of Camistat by looking at whether it decreases ATP levels.
Mm,
So again, they do high doses of Camistat to see whether it has any effect on the ATP levels because lowering of ATP usually equals like cell death or something bad's happening. So it's very basic, and again, there's not really much you can extrapolate from a cell apart from so they don't really try to look at more intricate things that are happening. Just live or
hmm
dead. And then
hmm
finally, they do use the actual like SARS-CoV virus and check with Camistat and infecting into some cells. So it would, and this one I feel like D isn't necessarily as convincing as I'd like it to be because it doesn't like knock down. So Camistat does like reduce the was it the genomes?
Yeah,
Sorry, the
the genome equivalents,
yeah, but it's not like
not
it
big.
knocks it down, but it's not like eliminating.
Yeah, but I
So
think it's also that's supposed to be suspected, right? Because in there's something else that can do the cleavage.
yeah, so that's fairly understand. I mean, they've only looked at two proteases that can do cleavage, but theoretically there could be more. But we're only looking at that in this study because
Hmm.
we're basically focusing on what is known with already known original
Right,
SARS.
right.
Uh,
But then also, yeah. Oh,
yeah,
inoculum. Oh yeah, 24 hours the inoculum. Yeah, there's something else happening, and
yeah,
and that could be because of another protein on the surface, right?
yeah.
Of it. Yeah.
Yeah, so because SARS has multiple proteins, so there could be some. I mean, for a lot of viruses, I think a lot of them do have their own cell proteases that encode. I'm not sure.
Hmm.
But again, with viruses, everything can happen, so I could point to
Yeah.
lots of things. So, um, and then in 4E, they look at two doses and looking at the cell entry for just looking at the effect of Camistat on cell entry for SARS-S and SARS-2-CoV. So I think this is where they come to the end of that Camistat story, just by looking at the big versus low dose. Um,
Yeah. So Camistat is I just try to want to reconcile again my thoughts about A and B on this. So Camistat is an inhibitor for TMPRSS2,
yes.
right? And adding it in prevents some amount of fusion from it.
Yeah.
Unless you overexpress TMPRSS2 on the cells, right? That's what's happening in A. They like put a bunch more in it, and
Uh,
then, or is it
I,
that Vero cells don't have any TMPRSS2?
yeah, I think with Vero cells they don't have TMPRSS2 and they have to add it in.
Hmm.
So that's why it
Ah,
knocks.
yes, I see. Yeah, because there is no effect of Camistat. Yeah,
Yeah. So
I
it's
see. Nothing is getting infected. Percentage of control is nothing's getting infected in the with the other when they add the other inhibitor. It's all based on the the CATB or whatever that's allowing for entry.
yeah. So
Okay.
I guess the idea is that the CATB does have an effect, but it's not that. It's not the same level of effect as so. It's a bit weird because they don't necessarily compare them side by side uh,
Hmm.
directly. So I mean it's I mean yeah. So it's I feel like
There's
they
something there's something there I feel that isn't
well
being said.
the thing that kind of baffles me slightly is that they did that original pseudotype virus on Vero cells where they didn't seem to add anything, but it still caused like an infection. Like that's all the way in the figure.
In the
So
first one, right?
yeah, in the first
In
one. So the question is like, what was going on there? I can't zoom all the way
yeah.
back, but yeah,
Yeah. The pseudotype entry, that's X fold over background. And
yeah,
then in this graph, it's percentage of control. So X fold over background, they are seeing small amounts, but the control is control background? No, control is the control is VSVG, right? When they say X% of control.
actually, yeah,
No, otherwise, no, that's not
uh,
true. Percent of control is not VSVG because otherwise that would all be at 100, right?
yeah,
That would lock it into 100 there.
yeah.
So percentage of control is background in this
Yeah.
sense, uninfected control.
So that doesn't yeah. So that's one thing that doesn't. So that is kind of confusing because they find two
Yeah.
different things. Uh, so I would be like kind of
Something's
one.
missing there. I feel like yeah, I missed that when I was reading this. I was like, there's something more to think about here.
Yeah,
Like some other meaning that should be coming through that, yeah, I don't think we've talked about yet. But
yeah.
anyways, as it is.
I get that's one of the things I'm not quite sure about what the deal is with
Mhm.
that. So, um, yeah, what's the deal with that thing? Those two things being different. Why would the Vero cells be? Why would the Vero cells behave so differently if they? Because
Yeah. Oh, I wonder if could it be inoculum? Could it be how much of the pseudotype virus that they add?
I mean
That
that
it's
could be very well the
two
case.
different two different actual assays that they're doing of those. Like they just have a different amount.
I think yeah. I mean it could be a number of things. So yeah,
Analysis.
it's weird because I've got this one thing that basically says like x fold over the back 10 to times 10 to the 4 over background based on um, pseudotype entry. So
Hmm.
um, yeah, it's so yeah they basically
Yeah,
had versus particles with no envelope protein, and then they've got suddenly Vero cells are no good at being infected when they have uh,
yeah.
so
The
it's
zero is so odd.
that is very odd. And again, you see it's a similar thing with the H293T, because again, like that,
Yeah, in the supplement, right?
yeah, in the supplement, that again is is a cell line that should have some good. Actually, no, sorry, yeah, no, that's right. Sorry, yeah, um, yeah, 293T again. That has some pseudotype entry, but suddenly it doesn't in the other figures. So that
Hmm.
is a very unusual effect to happen. And I'm not sure they quite clearly answer why that is.
Yeah. It must be something that they're doing in the experiments between them. It doesn't really say anything that they did multiple ways in the
Yeah,
methods,
the
but then
methods.
sometimes methods aren't always like there also might be the star. I know cells the star methods that have like the more in-depth ones afterwards,
Yeah,
so that's possible as
but even
well.
so, this method has heavy reference syndrome where they go like as done in previous studies, rather than
Right.
that's so I mean it's understandable because sometimes you want to cut down the size of the references, but it also makes it quite harder for people to find out what you've done. Um,
Hmm.
so again, quite a lot of these kind of the assays they do tend to be referencing uh, techniques that have been described elsewhere. So it's not necessarily clear what the difference in those pseudotyping experiments were.
Right.
So it's yeah. So
Yeah, I feel like they don't really tell us the amount of virus that was added into these experiments.
not especially. I mean I mean
At
it's
least
not.
it's not clear to me from reading the figure captions. I just like
Yeah,
ran through the figure captions and the methods. I was like, okay, we don't know how much they put on these cells necessarily of
yeah.
the pseudotype virus.
Because the way the methods are set out, they talk about the strategy where they do pseudotyping, but they don't talk
Well, they
about for each.
make the virus.
Yeah,
Yeah.
but they don't really talk
They talk
about.
about making it.
Yeah, they don't really talk much about each individual experiment.
Yeah.
So, so the methods are very generalized, which means that it is actually quite a trial to read through this paper because you have to understand what's going on. You have to be going back and forth through what they write in the text, what they write in the figure legends, and what they write
Hmm.
in the methods, because there is no repeating on anything here, which is both a very efficient way of, but they don't.
Very efficient.
It's very efficient because they don't waste any like kind of letters on anything, but that means that you have to track your way through the paper to try and find each individual point, which
Right.
can be quite time consuming. And even then, it can be quite confusing as well because
Yeah.
again they write things in a general way that means that you have to do a lot of like kind of make a lot of assumptions on what they've done.
Hmm.
So
Okay,
yeah.
sorry. Sorry for digressing. Let's do
Oh
the final
no,
figure.
talking shit about papers is what we should be doing. I mean, this paper was bad, and it's not because I didn't understand it. But yeah, no, actually, I mean, this last figure is almost comes out of nowhere. So,
Mhm.
figure 5 is almost like a figure from an unrelated paper out of something completely different.
Well, I guess again, they did show us before that there's antibodies that work to neutralize this.
Yeah,
Um, so they're just tacking on another one that might give it some more clinical relevance, I guess.
and yeah, I mean, for us, it's going to be a bit difficult to read this because we've seen papers that delve into this kind
Right.
of
So
in
deeply.
much more depth, whereas
Hmm.
in this study, it's very much surface level of does this neutralize a pseudotyped antibody, or whereas in previous we've looked at T cells, we've
Right.
looked at kind of the actual like kind of molecular structure, we've looked at vaccine studies, which really go in
Yeah.
depth in this. So this
Yeah.
one, they're
Like in some ways we can predict. We know that regular SARS will probably have their serum will probably have some effect
yeah,
because we've seen the epitope mapping
yeah,
of antibodies from those people. Yeah, actually onto this.
yeah. But I mean, even so, they do show that okay, well, there is some neutralizing
Yeah.
effect.
And again, this paper came out in April, right?
Yeah,
And like having these data points early, right, in all of this journey, the
yeah,
scientific research journey, like undoubtedly steered people in this direction as well, right? It's like more confirmation. Like other papers were confirming this. We're just reading this paper kind of late
yeah,
in
exactly.
the dialogue game, yeah.
So yeah, we
And the fact that they then also make rabbit serum against
yeah,
it is it sort of like the initial right is those initial like preclinical style trial right saying
yeah,
that look we can do like a vaccination and make neutralizing antibodies.
yeah. I mean, this is all good stuff, and if we were reading this in April, we'd be really happy about it. But
Yeah, now it's kind of like okay, we got tons of candidates. We've seen different constructs do this, right? This is
yeah,
just another construct that gives us this result. But whether or not that's going to make the leap right into humans from both the safety and the efficacy standpoint, huge open question.
yeah,
Yeah.
yeah. I think so. So I mean, I guess that's something I have looked into. Like, there is a Camostat clinical trial going ahead
Mm.
for coronavirus, and I know
Which is
that
nice because you said it was a cancer drug, right? So
yeah,
there's this idea that they're just repurposing something, right? They know certain effects that it has.
yeah.
They know what the side effects will be at some level.
Yeah, I think that, and the fact that they already know that safety profile means that they can jump straight into phase 3, which is quite
Hmm.
a good thing, and hopefully the results
Oh,
will
they
be
probably no, they maybe they'll do a phase 2, 3. Like they still have to do. They don't know the dose
yeah,
to give
true,
to make it work. Yeah. And
yeah,
the other thing is that it's oh right, yeah, because it could be used. That I mean, that's another thing. This idea is that it's blocking a development of viral load,
mm,
right? Like you can give it afterwards, and you won't get so much virus to come out. Is that important in the clinical course of SARS-CoV-2? Right, because the remdesivir, which blocks viral load but does it at an earlier stage, it's not like for the clinical
mm.
course, right? Like not during the infection. I wonder if this one is a bit different. I feel like they're trying to address that, right, with pre-incubation versus not pre-incubation of adding the intervention.
Yeah, I mean, compared to like, I mean, this is basically set up almost like an antiviral in the way it tries to block attachment, which means that this is probably going to be much
Hmm.
better at prevention stage rather than at the late stage treatment where you're mostly dealing with the immune system.
Right,
So,
right. And so again, like that would open up like another that in some ways that opens up phase 1 and phase 2 again because you're giving it in a really different way than you normally would, right? Like you want to see those doses. They may not have that information for those safety doses.
and
But
the,
did you say you saw a phase 3 of it,
oh no,
or
I
you
think
just saw a
I saw
clinical
a clinical
trial?
trial. I forgot what phase it was at. Uh,
Yeah, it's still maybe an early one. I
yeah,
don't know if they
so
could jump that much.
yeah, because again, this isn't working on a viral protein. This is working against a human protein, so it is going to. There is a likelihood that they'll cause a side effect because human proteins have stuff to do.
Yeah, yeah, exactly. Yeah, and I think that's like an interesting thing that comes out with cancer too. It's right, human proteins have stuff to do, but when you're dying of like some crazy chronic, right, crazy disease like cancer, it doesn't. There's sort of like the cost benefit it gets weighed, right? The classic one is chemotherapy, but like infectious disease prophylaxis, that's a little bit different because you're not. Yeah, the risk of that is just different. Like just don't expose yourself, right? It could be a really important thing
Yeah.
instead of like taking this drug all the time that gives you. Yeah.
Okay, so there's a phase 2A clinical trial and a phase 2/3 randomized multi-center trial
Yeah,
looking at
that
this.
makes sense. They because they know a little bit about the safety, but they still really don't understand the dosing aspect. They got to figure out an efficacious dose.
Yeah, I mean, the other thing I did see like an internet comment saying that from a doctor saying that that in patients who are treated for pancreatitis, where Camsat would usually be prescribed, their
Hmm.
ratio of kind of infected patients is lower. But again, observational internet
Sure.
comment, not going to take too much time to, but pointing out that
Yeah.
there is positivity out there.
Hmm.
I mean, I'd be kind of surprised at that because again, Camstat hits one of like two important proteases. So while I'd assume it'd like reduce it, I wouldn't assume that'd be like a cure-all
Right.
because
There could be other things that take over
yeah.
that cleavage.
So it'd be very hard for me to like say that okay, this is going to stop it because then there's always that CATBL thing that can happen. I mean, if there's a
Right.
combination of the of inhibitors, then I would be kind of interested in, but
Yeah.
but
But I guess the other thing is that in our list of potential selections, when I found this, it also led me down the path because I started searching TMPRSS2 in
mm,
the literature and being like, how is this being addressed? And there's all these small molecule panels that people are
yeah.
trying to do with the receptor and whatever it needs, right? Proteases and binding, yeah, and binding the spike itself, looking for things that inhibit that. So, like, this
Yeah,
is another area of the therapeutics that we haven't talked about that much. Like remdesivir is one of the examples, but really small molecules is also a whole other like
yeah,
realm of pharmaceutical interventions that could be that people have different ways of trying to figure out what the candidates are.
yeah. So I
Yeah.
think that it's fairly positive and kind of very interesting to actually get into that receptor interaction and get into like old
Hmm.
school like
Yeah, it's not just binding, right? Like I think through this whole time we've been talking about binding and the story is not just like it gets bound and it goes in, right? It's that it gets bound, the cell like puts it into an endosome, something happens in the endosome, factors occur, right?
yeah,
The spike gets cleaved and then fusion happens. There's all these steps.
yeah,
And that's like, pathogenesis in a
yeah.
nutshell,
That's
right? It's so easy to
yeah,
say it gets in, but there's so many things that happen along the way.
yeah. Figuring out how these tiny molecular machines work is kind of an interesting part of microbiology, and this paper kind of touches on that surface. And
Hmm.
like we could look into it in a bit more detail at some point, or look at other things because this is kind of how viruses work is very interesting to
Hmm.
microbiologists.
But you can see that even in light of like, I felt like we chose this to figure out like how things work, but even the light of this paper is skewed as like what the intervention might be.
Yeah.
Like it speaks to the papers of the time that are coming out now, right? It's like people want to know these things, and there's like a huge drive, yeah, to be very relevant here, to throw relevant information out, therapeutically relevant information out.
I mean, I feel like that happened in normal microbiology before, where if you have a basic microbiology paper, you can go to a higher impact journal if you can try and find some way to prove that it has a treatment effect. I mean, I know
Right.
lots of. I mean, there's an old joke where like what do you call an endocrinologist who gets paid? Someone who's found a way to turn that into an obesity drug. So a lot of the obesity studies were people who were like really interested in endocrinology but need that money and go, okay, right, well, we need to find some way to make it fit obesity, and
Related to
then
obesity. Yeah.
yeah, and then suddenly you'll get funded. And it happens a lot in times where you get these papers are really brilliant molecular biology papers, and then suddenly at the end they try and tie it to a treatment. And that
Yeah.
kind of felt very much in this way, where they did all this cool molecular biology, and then they said, okay, let's swing some antibodies in there and see what happens, and then we
Yeah.
can kind of boost and say there's treatment potential.
For sure.
Yeah, I mean,
Yeah.
yes, even science is susceptible to selling and marketing. Uh,
Yes. It just comes out a little bit differently.
yeah, to a different audience who are sensitive to different things, especially now where everyone is looking for the thing that'll get us out of our living rooms and into society again.
Yes, for sure. For sure. So yeah, that was a great discussion about this paper.
Yeah,
If you have suggestions about papers you'd like us to dive into, please tweet us using the hashtag or our tags, and we'll see you next week
yeah,
for news to try to choose the next one.
yeah. So thank you very much. We'll see you next week.